here. So it's great that you're all here so early on the last day. It's been a fantastic meeting, and kudos to the organizers for bringing us all together, and to the NIH staff who helped foster this field through the Tissue Chips program. What I want to do today is share with you first a little bit of intro to some things that have gone on in lab related to the topic of this conference. That'll be somewhat of a review and survey more than new stuff on the organs on chips, but then really dive into something I think is super important for this field, which is conceptualizing the biology in patients, particularly for complex diseases, as complex diseases that involve inflammation are some of the hardest ones to develop drug. Who's staying here for the American Diabetes Association meeting that starts today? Okay, I see a couple people like me who'll be here. And I'm going to focus on a particular disease because I actually go in the operating room and collect samples from patients, and I go to many, many clinical meetings in that space and explain how we deconstruct the patient, and then of course come back to tissue engineering and organs on chips. So that's the talk today. So first, the little survey. I became involved in this field actually at its beginning. I had my roots in tissue engineering, working with a transplant surgeon and Bob Langer. I was their first tissue engineering postdoc, and we focused on trying to build liver for regenerative medicine. But in the 90s, there was this aha moment that all of these things that patients were getting liver transplants for, we just didn't understand the disease very well. It was a complete dearth of models to explain human disease. So I shifted the focus of my lab toward building these kinds of in vitro models. And when Gail Naughton, one of the early tissue engineering pioneers, founder of the company Advanced Tissue Science, and I were asked to write a perspective for science on where the field of tissue engineering was going, we included in it that we really thought it would be in the future these model systems for complex diseases. So this is 20 years ago, and you always say in 20 years we're going to be, but here we are. In fact, this is a huge, huge, huge growing application of the field of tissue engineering. So at that time, we had developed a very simple 3D perfused model of liver involving a little silicon chip shown here on the left, deep reactive ion etching. You see cells into the scaffold. This is the basis of what CM Bio now has as a liver chip. There's a membrane on the bottom. The cells undergo morphogenesis. And in this initial iteration, they formed tissue-like structures. These happen to be rat cells. And when we combined the different non-parenchymal cell types, you could see features of some of the sinusoidal, but in no way is this liver. The value of this arrangement, though, was the perfusion and so on allowed us to get good mixing and keep the tissue functional for weeks at a time. So it was a lot of interest, collaborations with University of Pittsburgh and others, led to a lot of interest that led us to simplify the operation of the chip. Albert Waugh, who's here from NIH, struggled with this very first iteration on the left and published some very nice work with sinusoidal endothelial cells, but we translated it into a multi-well plate format, and critical to that was the development, together with my fabulous collaborator Dave Trumper in mechanical engineering, of onboard microfluidic pumps that are pneumatically driven. So these are just little pneumatic pumps that circulate fluid around and around, driven by air and vacuum. So there's a membrane between the top and bottom, and the fluidic channels are machined in this case. And so this has now been commercialized, and you probably have seen several of the presentations by CM Bio in their booth and so on, and they continue to be a wonderful collaborator and supporter of our work, and we actually order liver chips now from them. So since the beginning of my work in this field, working with pharma, going back 20 years, working with Cindy Afshari when she was at Amgen, all the problems that were brought to us in liver involved some aspect of immunology, idiosyncratic toxicity, et cetera, et cetera. And this is just an example of a project we did with Amgen to try to dissect what happened, dissect a phenomenon observed when tocilizumab went into the clinic. And it was observed that patients taking small molecule drugs like statins, the metabolism changed so much that the FDA put a warning label on it about that. And so together with Amgen, we showed that you could recapitulate this phenotype seen in patients by creating an inflammatory situation in liver where if you had immune cells present, and in this case, Kupffer cells, and then relieved the inflammation with tocilizumab. So this could be recapitulated. And again, this isn't all the functions of liver, but it's an illustration of the value of these MPS systems. And so now we continue to apply these currently in a really lovely lovely collaboration with Novo Nordisk to liver metabolic diseases, and then to some other applications. So for example, here at the ADA meeting, Erin Tabonian, who presented a poster last night on work she's doing to continue work done by a postdoc, Jessica Ungerleider, is investigating in vitro metabolic disease models here, showing that you can induce insulin resistance by hyperinsulinemia. As many of us have mentioned during this meeting, the use of insulin in cell culture is insanely hyperphysiologic for most cell culture. So that's an example. We, of course, were one of the groups that was involved in the big DARPA program, our group and Don Ingber's group. And we use this as an opportunity to expand the pumping technology to interconnect 10 organ systems together, living at one time in constant communication on a platform. So unlike the Emulate system where they pipette between, this one actually had interconnected living for a month. And Murat Chirit, who's here and now heads Javelin Biosystems, headed our translational systems pharmacology team, and he did all of the modeling for the pharmacokinetics such as shown here. So this was a demonstration project. DARPA let us continue demonstrating more biology. This just showed that you could do this, but then we applied the platform to some very interesting physiological situations involving gut and liver. And Martin Trapachar, who's very involved with this meeting, when he was a postdoc in the lab, led two really fun projects looking at gut-liver interactions and gut-liver-brain, specifically around how short-chain fatty acids may be influencing physiology in inflammatory situations. And I'm not going to show all the data, but the punchline in this story was that they're a double-edged sword in health. They can be very beneficial, but when there's inflammation, you can get a potentiation of certain deleterious effects. And you can read his work, and he's here at the meeting, and you can find him at the break if you'd like more on that. What's stimulated us since the DARPA project, though, is the need for much more sophisticated tissue engineering systems to put into these platforms. So we first showed, we extended our platform technology, again, not with great tissue engineering, but that's what I'll talk about mainly in the talk. We extended it to show that you could culture one of the most fastidious oxygen-sensitive microbes, Faecalibacterium prausnitzii. Sorry, I'm getting used to this. Which is a super strict anaerobe. This organism is responsible for production of a lot of the short-chain fatty acids, particularly butyrate, in your lower GI tract, and it dies in the presence of oxygen. So this is a continuous culture, human colon monolayer here on a transwell, and the bacteria growing here. And this has also been published. So this demonstration showed us that we have the technologies to do this, but we have a monolayer here, so we need a much more sophisticated model. So what I'd like to do now is turn to ways that we think about deconstructing these complex diseases so that we can build MPSs that will help us discern the path forward for drug development. Who doesn't know about endometriosis? Oh my God, I'm so happy everybody knows, because 10 years ago, that wasn't the case. So it's a chronic inflammatory disease characterized by ectopic growth of endometrium. We don't know what causes it. It could be developmental. A lot of young girls have problems starting right when they start their periods, but others develop it later in life. There's many different forms the lesions take. Here's some in the abdominal wall, highly vascularized. This is invasion into the colon. The thing about them, they're beautiful cystic-like structures. They retain a monolayer epithelium, completely polarized. So it's not like a tumor where you get all jumbled cells that lack polarization. And of course, it can even invade through the diaphragm and get to the lung. And this is very serious. Patients really suffer. Super prevalent. And if there were good treatments for it for everybody, I wouldn't be talking to you here today, right? So about 30% of patients have invasive disease. This is a patient of my collaborator, Maurizio Brau in Sao Paulo. He had to take out 10 centimeters of this patient's colon because she couldn't poop. Okay, so this has many manifestations and has remained clinically very mysterious. The landscape is very murky because the symptoms don't correlate well with the staging of the disease. So a patient with advanced disease might have very few symptoms, and a patient with one lesion may be crippled in pain and unable to work during her menstrual cycle. Who has heard of adenomyosis, or who has not heard of adenomyosis? Okay, so adenomyosis is the sibling, if not the twin, of endometriosis. It's when you have these lesions growing in the muscle of the uterus, the myometrium. And so you can see, again, beautiful epithelial structures sitting right there in the muscle. And about 1 in 10 women are estimated to have adenomyosis. To give you a sense of research intensity in the field, let's compare we like to use PubMed as a surrogate for measuring research intensity in all research sectors, industry, and around the world. And if you look at Crohn's disease, which afflicts about 1% of the US population, there's over 60,000 papers published. Adenomyosis, which probably afflicts 10% of reproductive age women, if you normalize, it's probably afflicting at least twice as many US citizens as Crohn's, and yet there's only 3,000 papers, 5% of the research intensity. NIH only has 2 grants ever on adenomyosis, and one of them just started. So this is a really huge problem because these diseases are under the radar despite afflicting a huge percent of our population with crippling symptoms. People miss work because of this, seriously, and they can't get pregnant and all these other kind of things. So when we came into the field in 2009, I got pulled in by a gynecology surgeon, and for some personal reasons as well, including the need to be thinking about how we could apply engineering to women's diseases more prominently. What was striking to us is that the disease was treated as binary. You're a patient or you're not a patient, and you have stages that are staged according to disease burden. Stage 1 is a few lesions, stage 4 is a lot of lesions and endometrioma. But there was enormous heterogeneity in the clinical landscape in terms of symptom presentation, types of lesions, et cetera. And so we started to question whether the staging, which is just disease burden, really made sense. And so we promoted the idea that it's not one disease, that perhaps there are molecular subtypes that one could identify. And so we started out about almost 15 years ago now to deconstruct the patients. So to move beyond this idea that the lesion burden is a classification scheme. And a miracle happened right after we got a foundation. We started with a foundation grant of about $1 million. And right after that, I got breast cancer. And it was incredibly, incredibly illuminating because within days of finding a tumor myself in Singapore, do self-exam, women, I was classified as having something called triple negative breast cancer. So the 3 molecular markers that were related to the mechanism, prognosis, and therapy for the disease immediately based on clinical studies with thousands of patients. There was nothing like this in endometriosis. And even though there are not somatic mutations that are credible yet, we speculated that we might be able to find different subclasses of patients on a molecular basis. So how do we do that? And why do we do that? So to kind of orient you, in an era of personalized medicine where you might think every patient is different, and indeed every patient is different, there are probably subgroups of patients, maybe 5, 10, however many, such that you're not disease or not, you're not binary, but you can't possibly, with almost 200 million women around the world suffering from these diseases, you can't possibly have a different therapy for every patient. So we like to think of subgroups of patients that may be in the order of a dozen or so. And this is going to be a many decades long quest, of course, to figure this out. And so we embarked on what we call the systems biology of endometriosis, where we study the disease in the eutopic tissue, the endometrium, where you have epithelia communicating with stroma and immune cells, in the peritoneal cavity where you have fluid that has about 1 million cells per mil, mostly immune cells, and obviously all their signaling molecules, and then the lesions themselves, which I'm showing here, are on the abdominal wall, and you can glean facets of disease processes from studying all of these tissues in a comprehensive way. And we've published a lot on this. I'm going to give you an overview of a couple of studies to give you a sense of how we approach this. So when we started, we had said, well, if there's not somatic mutations that you have in cancer, maybe we can glean insights by looking at cell-cell communication networks involved in the immune response and in the invasion response. And biologists had described a lot of inflammation markers in the peritoneal cavity where the action is, but they typically would focus on a single pathway or a favorite molecule like RANTES. But as engineers, we come in and think, how is the whole network operating and how are networks communicating with other networks? So this idea that we can think about information flow in an ecumenical sense and measure activity across the entire network to discern its function and its dysfunction, that was new. So we developed a couple of studies probing the networks, and one was the cell-cell communication networks involved in inflammation in the peritoneal cavity. And the peritoneal fluid is super easy to harvest. We've taught surgeons all over the world how to do this because you can get it right at the beginning of surgery and it's discarded tissue, so very easy IRB. You get the fluid and then you can separate out and save the cells to examine later, and then study the signaling molecules. And so we developed a way, and this was back, 10 years ago, it was hard to do quantitative Luminex. It's still hard to do quantitative Luminex. So we spent a lot of time developing a way to do Luminex to get quantitative information on about 50 of these cell-cell communication molecules between the immune cells. And then we did an unsupervised multivariate analysis mathematical method to ask the question, are there groups of patients in whom changes in the immune network are correlated? So in other words, if you might have patients in whom some things are up and some things are down, and these patients are changing together. So we used a kind of math. It's the same thing that Netflix has traditionally used to tell you what movies to watch. And it's unsupervised, meaning we didn't give it any clinical information. So we didn't say who's a patient and who's not. That doesn't mean it's hypothesis free because we had a hypothesis about the communication networks. But we could then ask, are there groups of patients who are together who may have a common mechanism? Now, we did this because typical univariate analysis is not that informative. And this is a typical kind of result showing IL-8, which is always in most studies reported up in endometriosis patients in the peritoneal fluid. This is a log scale, and you can see the problem. Here's the controls, here's patients, and this is plotted two ways. These are patients who are taking a drug. And you can see the spread here. A huge fraction of our patients are overlapping with the controls, no matter how we plot it. Here's by disease stage, here's by menstrual cycle phase. So there's always this huge overlap, suggesting that there's some patients who are different. Another way to look at this, and this is a different study, I'm not going to go into it, it's one we did recently. This happens to be endometrial biopsies. But again, you see the same overlap. This is IL-15 and endometrial biopsies. And this is all published. This has actually been published, sorry for the slide. But the one that came closest to significance shown here, so these are all the cytokines, IL-15, didn't turn out to be significant after we corrected for multiple hypotheses. But you see the problem here. Almost all of the data are overlapping with the controls. So now, though, if you go in and you add up all of the different cytokines, almost all of them have these features, but you can imagine that patients who are up here may have some other cytokines down here differing from controls, and now you get the power of multivariate analysis. So when we did that, we had an initial study with about 100 women, and we did this analysis, and what we found in about a third of the patients who had all the disease stages was a signature set of cytokines shown here that they all expressed, and none of the controls had this signature set. We've replicated this for the top 5 cytokines with Maurizio, and that paper hasn't been published yet because the postdoc left precipitously, and I've got to write the paper. But we could take these data and reverse engineer this immune network because immune secretion profiles are beautifully documented in many databases, and we identified a particular state of macrophage in the peritoneal cavity of these women and then experimentally validated it. And we also predicted that Jun kinase was regulating production of these. And this was the first time that Jun kinase had been implicated. And in fact, we showed that Jun kinase was by inhibiting Jun kinase. And then we did a lot of other kinase inhibitors. And so this was very exciting, especially because we then also implicated Jun kinase in a companion study looking at invasion with a similar kind of network analysis. And this involves in vitro immune invasion assays. And of course, this has also been published, so you can go back and look at this with a lot of math in that. And this was in collaboration with Doug Laufenberger, our systems biology collaborator. Now, what was fascinating, when we published these papers, almost immediately, we learned that Jun kinase inhibitors had cured multiple patient populations of endometriosis. Woohoo! Okay, however, they were not human. And in fact, Steve Palmer, the lead author here, was at Merck, and he had gotten Merck to sponsor 2 preclinical studies that then Merck decided not to move forward into the clinic. They licensed it to Preglem, their molecule, because Jun kinase inhibitors were being developed for other applications. The clinical trial was unsuccessful for a lot of reasons, probably not having to do ultimately with whether inhibiting Jun kinase would actually help patients. Issues with the drug, issues with the specific composition of the drug, issues with the trial design, et cetera. And so in talking to a lot of people, pharma, we got to have better efficacy models. So that brings us to how we reconstruct the patients and the topic of this conference. Okay, so now we want to go in and we want to build avatars of the patients. We want to capture key disease phenotypes in vitro so that a company can feel confident that if we propose a mechanism because we went in and studied all these patients, or they propose a mechanism because they went in and studied patients, that we can test that mechanism in vitro. And we can also obviously use many of the approaches here at this meeting to look for toxicity and other effects, because our lab has shifted mainly to analysis of disease processes, and we're not really hugely involved in toxicity studies anymore. And so to go in and think about this, obviously the lesions are there. Patients with no lesions do not have these symptoms. So how can we think about what might we do for the lesions? And it's a dream someday that we would diagnose this at birth and prevent lesions from ever happening. But we started thinking that surgeons will take the lesions out, but there are micro lesions still left in these patients. And are there ways that we can affect their growth into these more macroscopic lesions that are causing the patient so much distress? So part of the lab, I have a really dedicated postdoc who's going to be joining the faculty at Tufts in August in bioengineering, Juan Necco, and in collaboration with a local company, did some whole mount tissue imaging using the clearing process and light chain imaging to characterize some of the features of the lesion. So this is an adenomyosis lesion in situ, and this is looking down inside the lesion. Recall that these are beautiful cystic-like structures with a monolayer epithelium. And so you can see gland-like structures pointing out here, here, here. These are incredibly innervated. They're incredibly vascularized. And what we are doing is saying, okay, if we're going to recreate these lesions, we're going to think about the parameter space that's biophysical and biochemical that direct the properties of these lesions, because some lesions are very close to peritoneal fluid, which has an insanely weird composition of hormones and et cetera, but they can then invade into muscle. How crazy is it? You never even see carcinoma in muscle, but these are in muscle. So a lot of interesting features about the environment. So we create a parameter space, and we want to march through parameter space and understand how these cues are dictating function. So of course, we have to first build a tissue bank from biopsies from well-phenotyped patients, and then rebuild the patient. So how are we doing this? We had to develop an enormous number of new tools. And I think these tools will be generally useful to the community, and we're trying to commercialize them right now. A huge innovation was not ours, it was Hans Cleaver's. You can make organoids, and everybody here knows about organoids. This is an intestinal organoid, but it changed our world when this technology was translated into the endometrial space by 2 different groups. And we work with the folks in this group here, Hillary Critchley and others, because now people all over the world use these approaches to make endometrial tissue banks. Before this, we would get endometrial biopsy samples and the epithelia had to be a one-shot. You use it right away. But now we have a huge tissue bank with a lot of patients. So the general process when we started, of course, was to take the glands, isolate them, and build the organoids in matrigel droplet culture, as is standard, using the cocktails of growth factors. But as we know, matrigel is a huge problem for so many reasons. And we needed to get away from it. We also had a super practical reason for having to get away from matrigel. In the DARPA project that we had, tissues had to live for a month. The endometrium was one of our tissues, and there was no way you could keep a culture going for a whole month in matrigel because it disintegrates. So we had a very practical reason, which is that 28 days is a long time. So building on pioneering work by Jeff Hubble many decades ago that the universe has adopted, we did a very, very customized approach to using multi-arm PEG modified with cell interacting peptides, cross-linked with peptides. You combine these two precursors with cells, and we typically do a Michael addition, which is a small pH change, and you can create these beautiful gels that encapsulate cells very, very safely. And we published almost everything on this. You can find it online, so I'm going to do a whirlwind tour of it, but hopefully repeating some things you may have seen at some of our posters, but giving you enough information that you understand what we did. So a crucial thing for us, of course, is we want to build the entire lesion, and we actually also want to build the endometrium, because the endometrium is so fascinating, not just for recurrent pregnancy loss, but for regenerative medicine purposes, because it grows so incredibly beautifully without scarring. So what did we do that was special? The thing that our application drove was that we had to have epithelial cells be very happy, and they're very finicky. They want a basement membrane, and then stromal cells and immune cells at the very least, obviously endothelial cells and so on. So how can we build a matrix that satisfies all of these cells and is so-called one size fits all? What we did was, of course, cross-link things with a degradable cross-link so cells can remodel, and that is a very common approach. But we thought a lot about the particular adhesion ligands that went into this gel, and we envisioned it as you initially put cells, they're naked because they just got taken out of a tissue or they just got taken out of a cell culture expansion format. So they're naked, they have no ECM, they need some signals immediately when you put them in or on this gel. So we needed some minimal peptide adhesion ligands in order to engage integrins immediately. But then cells make their own matrix. So we have two ligands that we include, and this was based on a lot of engineering, including by Victor Hernandez-Gordia, who's here. He's now at United Therapeutics, and I'll show you some of his work in a couple of slides. But we had an unusual RGD ligand. Now, a huge fraction of the biomaterials community makes materials with simple RGD ligands, or maybe a loopy RGD ligand, cyclic or something. But that's partly because almost all cultured cells used in the biomaterials community express alpha V beta 3. Either they're tumor cells or they've been cultured a long time and they evolved to interact with vitronectin, fibronectin, and serum. But most primary cells do not have alpha V beta 3. It's very rare in the body. It's in tumors, wound healing, and actually in the endometrium for a tiny window during implantation. But it's not a real physiological ligand. It's a kind of wonky integrin. So we include the synergy site from the ninth domain of fibronectin. So here's fibronectin, so that we get a broader RGD integrin binding set of integrins. And this works very well with primary cells that are not expressing alpha V beta 3. And then a motif from collagen that can interact with alpha 1 beta 1 and alpha 2 beta 1. But that's not all. So that's to get the cells going. But importantly, we then also add these peptides that bind to matrix that cells produce. And that's because PEG itself is anti-adhesive. It's a blank slate. If cells are making matrix, it's not going to stick to PEG very well. So we make it stick to PEG. And that seems to work very well. And we published a huge number of papers on this by now. And I encourage you to look them up if you're interested in this. But let me tell you what we're doing with it. So when we started this, of course, we became interested not just in the endometrium, which is a really fascinating mucosal barrier, but the intestine. And there were many reasons for this. One, of course, was our DARPA funding required that we build all these organs. But going forward, there's also other reasons to continue working in the intestine, and they have to do with the funding out there. You can almost open up your shoot in your ceiling and get funding for intestine, but not so much the endometrium. So this is for the young people in the audience. But there's also fascinating intellectual reasons to compare these two together, in that in the gut, as we know, the stem cells reside in a crypt in a healthy, normal homeostatic gut. In the endometrium, in contrast, you have the basalis, which is always there. The functionalis is shed every month in cycling women. But you have evidence from several groups, including a recent publication from the Cambridge group, that there are stem cells actually out here in the lumen. Okay, so they're getting rid of, maybe they're what's causing endometriosis, right? So there's super fascinating questions about structure and function in these two mucosal barriers that we're pursuing, because there is evidence of stem cells here. And so we continue to digest those, and I don't have time to go into all of that fascinating biology in this talk, because I want to tell you a little bit more about the matrix, which was developed by Victor. And he took the scanning, semi-empirical scan of parameter space, thinking about the nature of our chassis, meaning the peg, thinking about the integrins that are in the crypt and how do we target them when they're naked so that they'll live and prosper, and then deposit matrix. And in doing this, he identified a composition of hydrogel, completely, totally synthetic. So you're not going to go in the supplemental information for this paper and find that we doped it with laminin. There is no doping of laminin in this, as there are in some other papers, and it's very confusing sometimes to figure that out. So this is completely synthetic. So here's cells growing in matrigel, and this was done multiple donors from the upper GI as well as the colon, that you can get clonal growth from single cells in this, not with the efficiency that you get in matrigel with this initial formulation, but we've done a lot of work since then. So a crucial thing was his figuring out that you needed active Gefoger. If you substitute this crucial residue in the motif with analog, you don't get growth of the organoids. And that's all been published. We're now exploiting some fascinating properties we observed with this to try to generate true mucosal barriers and barrier function by having fusion of the organoids with the surface. And this was a poster that Elise Gubbins in the lab presented last night. And again, this is one size fits all. So you can include stroma and immune cells, and the immune cells can traffic through this and so on. So let me finish up by explaining what we've done with the endometrium here, because I have plenty of time to finish what we do. Okay, so we want to come back to the endometrium, which again is really intellectually fascinating because more than other tissues, it is dramatically, well, except for the ovary, of course, and other reproductive tissues, it's dramatically influenced by the cycles and hormones. So typically you start your period and the functionalis is shed, and then you're going to grow the endometrium back. This is driven initially by estrogen and what's called the proliferative phase. So there's this massive centimeter big growth of the endometrium. And then ovulation occurs and progesterone is produced by the corpus luteum. And you get a maturation of that endometrium. And the stromal cells, in the fibroblast in the endometrium, bifurcate into different populations. Some remain responsive to progesterone and they become epithelial-like. They make a basement membrane, they produce prolactin, they make gap junctions and cadherins, and they look very epithelial-like. And then another population of fibroblasts becomes acutely senescent and makes matrix and growth factors and so on. So some fascinating biology, and you retain along the way stem cells in that fibroblast population, as shown very beautifully by Carolyn Gargette's group in Australia. Okay, so you have the secretory phase, and if you do not have implantation of an embryo, progesterone falls precipitously, triggering a cascade of events that causes tissue breakdown. So you've got this whole approximately 28-day cycle, and it's a challenge in tissue engineering because it's a kind of long time to have the tissue be resonant. So let me now take you through some things we're doing in tissue engineering and introduce you to one concept we use to characterize the phenotype. And so even in 2D culture on plastic, as shown here, these are stromal fibroblasts from the endometrium. They have a very unusual phenotype. They express huge levels of CD10, a cell surface endopeptidase, that's also expressed in the immune system. If you give them a progestin, they undergo this process called decidualization, in which they turn into those epithelial-like cells, and they make buckets and buckets of prolactin, a molecule involved in many physiological processes, and it's broken down to create submolecules that are very involved in regulating angiogenesis and endothelial function. Many uterine disorders, including endometriosis and adenomyosis, are characterized by something we call progesterone resistance, which is one of those syndrome things. It means that if you give these stromal fibroblasts progesterone, they're not going to do all the things they normally do, including they're not going to make prolactin. Okay, so we can use that as a characteristic in vitro. And everything that I'm doing, this is all patient-derived cells. We no longer work with cell lines. Okay, so as one example, and this is all led by Juan Necco, who's transitioning to his own lab this summer. And let's focus first on those fibroblasts. And in 2D, I just showed you what they do with giving prolactin. So you have them in estrogen, you give them a progestin, and we're using methoxyprogesterone acetate because progesterone is metabolized very rapidly. We're transitioning to progesterone, and I can talk to people about that later if they want. But you can see that the bump up in prolactin production after a couple of weeks, which is a characteristic experimental protocol, is modest. But if we put them inside this 3D gel, the response to prolactin is progesterone is dramatically enhanced. And this is true across multiple passages, et cetera, et cetera. And I'm not going to show you the data, but if we culture these cells on this gel, and this gel is about 250 Pascal, 250, 300 Pascal, so similar to the endometrium. If we put cells on top of this gel, guess what they do? It's crazy. They act just like this. So even a soft gel does not generate this. So this is an area that we're still investigating. There is a paper in preparation. We're not fully understanding why the soft gel 2D culture doesn't help that much. Okay, but let's move to building co-cultures of the endometrium, because we really have enormous crosstalk driven by these hormone cycles, crosstalk between multiple cell types. Let's just first look at the epithelia and stroma. In fact, I'm only going to describe epithelia and stroma, even though we're doing a lot with the other cell types. So first you can combine the stroma and epithelia. And here we're just using organoid culture. So you could see the beautiful apical actin, which is how the polarization should be. And if we were to stain for basement membrane, you would see it there. But these are the stromal cells interacting with the epithelia. And if you go through the cycle, you can recapitulate features morphologically that you would see in vivo. Now, of course, we want to do disease modeling. So a simple first characterization is add an inflammation cue to the culture media, IL-1 beta. And this is on bioRxiv, and it's going to be submitted any day now in a highly revised form from what's there, but the data are still pretty similar. So if you add an inflammation cue, you immediately see that these cells flatten out. They get about half as tall. The stroma become activated, and they do a lot of crazy things. And this is true across the whole menstrual phase. And there's a lot of effects on proliferation and so on that are described in the online version. And of course, adding IL-1 beta will, if you add IL-1 beta to control co-cultures, they make in this gel, they make a lot of prolactin, the controls make a lot of prolactin, but IL-1 beta blunts their ability to respond to progesterone. So this is the progesterone resistance that you see in patients. But importantly, the use of a synthetic hydrogel lets you to recapitulate features of the biophysical environment of the invasive lesions. So here we have a lesion. This is in the myometrium, and it is so crazy. These things are invading in the muscle. This is just wild. So this is the epithelia. This is by light sheet imaging that Juan did. And you can see these jagged edges invading into the muscle, and it is pervasive throughout the endometrium. Okay, and now if we create our gel to have the stiffness that I just showed you, but also to make a stiffer gel like that of the myometrium in an adenomyosis patient. And now we do these co-cultures. You can see these are beautiful, normal looking organoids, but in the stiff matrix, they take on features of the invasive phenotype. And there's some other behaviors that they engage in that are indicative of inflammatory just induced by the biophysical microenvironment. So now we have a number of tools to investigate this in a systematic way. One thing we're also doing is trying to compare to in vivo, and this is a little side story from a collaboration with Hilary Critchley's lab in Edinburgh. And Hilary Critchley is a clinician scientist. She's one of the leading lights in the world to investigate a phenomenon called heavy menstrual bleeding, in which in Europe and the UK, patients are giving a selective progesterone receptor modulator, and that's PRM. And the common one is ulipristal acetate, okay? And so you can have progesterone as an agonist of the receptor, and mifepristone, also known as the abortion drug, is a complete antagonist, and UPA sits somewhere along here, so that it interacts with the PR, but in a weird way, such that in the intact endometrium, it downregulates proliferation and but causes enormous morphological changes in the endometrium. And in the US, there was concern that these changes might be associated with later development of endometrial cancer, which is on the rise in the US. Of course, endometrial cancer is hugely associated with obesity. So women in the US are not able to take this actually super effective, wonderful drug because of lingering questions about what it may be doing permanently to the physiology of the endometrium. And so what Alex Salova, one of Hilary's students, did in collaboration with our lab, she decided to, Hilary had a patient cohort who were taking the drug, and she could take endometrial biopsies from the patients before they took the drug, while they're taking the drug, and then at a defined time after they had taken the drug. So this is a really valuable clinical cohort who had taken the drug and for whom we could not only study the molecular features of their endometria via the various omics, but we can also build the tissue banks and do the in vitro models and ask, are the in vitro models capturing the signatures that we see in vivo of these patients? And the huge question for us was, can you recapitulate, or can we understand more something about the proliferation of these cells and what is going on, because there's features of both proliferation and differentiation. And so could we illuminate things in a way that might get this drug more favorable light in the US? And so what Alex did in her thesis, it may sound simple, but it's really quite tricky to do these cultures, is ask, does the matrix composition affect the cellular response to this treatment in vitro? Okay, so she's going to look individual cells, stromal cells, organoids cultured separately and combined together and compare those. And she's going to do this in Matrigel and in our completely, totally 100% synthetic PEG gel. Okay, so here's a picture of them. Now, she won a big poster award at the Society for Reproductive Investigation in March, top 5 posters, and she got one of them, which was great because she had been a very not confident student going into the study, and she emerged very confident. This paper is, she just defended her thesis, and we're getting ready to submit this paper. So the punchline is here, and I'll walk you through this busy slide to tell you what the important thing is. Okay, so there was a lot of omics data from the patient samples in vivo from which we could glean that the proliferative pathways, the cyclins and all of these things were downregulated. So all the pathways related to proliferation were pretty much downregulated from the in vivo omics in the patient samples. So we wanted to ask we didn't have, enormous number of samples in vitro because this was all done during COVID. So we used a reverse-phased protein array of 100 different antibodies, picking the particular genes from the in vivo omics data. And so these were mostly cell proliferation genes, shown here in red, and decidualization here in yellow, and other proliferations in green. Okay, so the punchline is here. Okay, so these are the cells treated with estrogen. So they should be proliferative here. Okay, so based on in vivo, these are proliferative. And we see that in our PEG-grown gels, the genes associated with proliferation are pretty much upregulated, they're red. But everything associated with proliferation is downregulated in the PEG gels. Okay, so these are co-cultures. Matrigel is a mess. Okay, it's a mess. You're not recapitulating what happens in vivo. And these are co-cultures. So an interpretation that we're continuing to develop is that in the PEG gel, it's a low noise environment. Matrigel has all kinds of signals in it. So if the epithelia and stroma are trying to talk to each other, it's really hard because there's a lot of stuff going on in the way. It's like trying to have a conversation if you're on the runway at Logan and jets are taking off. You got to really scream. Okay, so only a few things are going to get through. Okay, but here, there's nothing in between. It's a quiet cafe, cup of coffee, bottle of wine, whatever, and the cells can talk to each other perfectly. Okay, so that's where we are. And so she then went back and looked at some of these things in vivo, and you always have to go back and look at the protein level in vivo by IHC with different antibodies, et cetera, and showed that the things that we were seeing in vitro were recapitulated in vivo. So that's hopefully to be submitted soon. Some of the clinical part of that has already been published. Now, I'm not going to go into this story, but I just want you to know of the publication. We had another collaboration with Klaus Jorgensen's lab in the UK on pancreatic tumor where we grew stroma and immune. These are just some data from it. It's the same gel. And an important feature of this is his lab did this all on their own with our instructions, and they made the gel themselves. So it's a very easy to use gel. And Klaus had worked with another lab, Matthias Lutoff's lab, and could not get the gel in that case to work, but found this one very easy to use. So now let me finish up with how we're modeling the birth of lesions, because what we really want to do is look at the lesions interacting with microvasculature. So taking a cue from the work like Chris Hughes has presented so beautifully over all the years at the NIH meetings, making microvasculature and microdevices, and then adding other tissue types to look at their interactions with the microvasculature. And of course, our big question is, we're giving patients a lot of drugs that manipulate hormones with the idea that somehow we are suppressing production of estradiol, that if you give a patient a menopause drug, this is a huge market for menopause drugs, that it's going to shut off estrogen production. It turns out that's not the case. A lot of patients actually have progression. I actually had progression on a GnRH analog, and it was terrible because I had terrible menopause symptoms, but I grew a lot of lesions that were very painful. So we want to understand what's really going on in a lesion, because steroid hormone metabolism is really, really wonky. And so we are working with Roger Cam, who's local, who has technology similar to Chris Hughes. And you've probably seen a lot of Chris's work here at the meeting. It's incredibly beautiful work and very inspiring. And this involves putting the endothelial cells and fibroblasts into a gel, typically a fibrin gel, in the center of a device like this. They form networks, and then you can flow cells through these and look at recruitment and so on. Now, Roger's lab has used this for a lot of applications that are published, many, many scores of applications. For example, looking at how tumor cells extravasate into extracellular space. So the tumor cells here in green, neutrophils are helping them move out. Once they move out into that space, you can watch them grow because these are devices you can do imaging in. And he has commercialized these devices. The AIM chips are available, and a lot of people use them. Their website is pretty helpful. And this is our student, Ellen Kahn, who gave a talk on Wednesday morning on how she's adapting some of these approaches to the endometrial models under our tissue chips U01. And this was her first attempt to recapitulate things that have been done in Roger's lab, building the vessels in the AIM chips and flowing monocytes through them. Okay, now for the lesion model, what she's done and what she presented at this meeting is there is a CAM lab iteration where devices that are now in a prototype stage in PDMS, you form the vascular bed and you leave these little holes and you can put tumors in. So we've been trying to build the endometriosis lesions in, and what Ellen presented was the vascularization into a proto-lesion from a preformed vascular bed. This is fibrin, and this is all PEG because we have to use PEG to grow our lesions. So we're hot on making a synthetic matrix that can be vascularized, but for now, this is the mode we use. And there are ways you can use PDMS for these kinds of experiments with hormones, but you have to be very careful because the hormones, you must saturate the devices with the hormones first, and you can't cycle anything in PDMS. So that, of course, has prompted us, and we're very grateful to NIH for 2 grants that support our work translating all of this into thermoplastic chips that have embedded pumps of the sort that drove our liver chip and drove the DARPA project. And this is not a story for today, but this is a photo of one of the prototypes. It's not the exact one we use, but we're using this for many applications in liver, gut, everything, and I'm going to be done in 1 minute. And so I'm finishing up. We actually are starting a consortium at MIT to translate these things with Roger and Ron Weiss. So if you're interested, if you're a pharma company and you're interested in learning more about that, you can ask me. But this is my last slide. We can finally test our Jun kinase inhibitors. We're teaming up with Steve Palmer, who went into academia. He's developed new variants of inhibitors. He's a really great medicinal chemist, and we're going to test these together in these new devices. And so that's it. Thanks very much.